Hello my dear students, this is Dr. 𝗲𝗵𝗵𝗺, your biochemistry faculty. In this video, we are going to discuss about a very important topic, vitamins. So let's start with our discussion on vitamins. We all are familiar with this term, as we know that vitamins are what? They are the nutritional supplements which are required for the normal growth and maintenance of the body, or I would say proper growth and maintenance of the body. Right? Now see, always remember, vitamins are basically the organic compounds. Organic compounds means they are the basically compounds formed of carbon. So they are organic in nature. So they are the organic compounds. And these vitamins, this was coined by the scientist Funk, and these vitamins are of two main types: water soluble and fat soluble. Water soluble, and it is clear from the word itself, water soluble means they are soluble in water so that they can be excreted in urine easily, and fat soluble. Fat soluble means which are soluble, which have the nature just like fats, and we know fats are strictly non-polar in nature, right? So if we talk about the water soluble vitamins, they will comprise of vitamin B complex. Now why vitamin B complex? Because vitamin B consists of a series of vitamins along with vitamin C. And if we talk about fat soluble vitamins, vitamin A, D, E and K. These are the four fat soluble vitamins. Okay. Now coming over to this, what are the major differences between water soluble vitamins and fat soluble vitamins? So if we talk about the major differences between them, because now we have to concentrate on the biochemical reactions. So the most important difference between a water soluble and a fat soluble vitamin is that they are always used as coenzymes. Very, very important. The B complex vitamins are mainly used as the coenzymes, whether we talk about decarboxylation, whether we talk about oxidative decarboxylation, whether we talk about transamination. So, B complex vitamins will be used as coenzymes, whereas under fat soluble vitamins, only vitamin K functions as a coenzyme. So this is the basic difference. The rest are the very common differences. I can say they are not stored in the body. So once their function is over, they will be excreted as such. If we talk about fat soluble, they are always stored. And whenever they are required, they will move to the target sites. The most important thing here we have to remember is that since they are soluble in fats, so they will always require the help of chylomicrons to be transported from one side to another side. So obviously if there is anything wrong with the fatty acid digestion, it is going to reflect the vitamin which are fat soluble, and that is what exactly happens when we discuss about A-beta lipoproteinemia, where the main MTTP protein is absent, the gene coding for MTTP protein is absent. So there these fat soluble vitamins are going to suffer, and the infant suffers from 𝗲𝗲𝗵𝗿𝗺. There will be excretion of fats in the feces. Then they can be excreted in urine easily. When their function is over, they are water soluble, so excreted in urine. They are never excreted in urine. In a normal individual, never excreted in urine. Right? So these are the three main differences between water soluble and fat soluble. Or fourth, I can say no chylomicrons are required. But if we talk about this, chylomicrons will be required for the transport, right? Then after this, then another important point about vitamins to remember is that no doubt we get vitamins from the outside diet. Whenever we are suffering from any disease, okay, vitamins are given as a supplement, but there are certain vitamins which can be synthesized in the body, and those type of vitamins are known as endogenously synthesized vitamins. So what are the endogenously synthesized vitamins? Synthesized vitamins are, first of all, if we talk about by the body. So if we talk about the endogenously synthesized vitamins by the body, niacin or B3. Niacin, it is the most important endogenously synthesized vitamin by tryptophan. And we know tryptophan is an essential amino acid. So 60 milligram of tryptophan is required to generate 1 milligram of niacin, right? Then there are certain vitamins which are synthesized in the body, and the vitamins which are synthesized in the body, they are generally by the intestinal flora. So we can say that the vitamins synthesized in the body. The most important one is, you can say, vitamin K, very important, vitamin B5, okay, and vitamin B7. Vitamin B5 is again pantothenic acid, right? Vitamin B7 is biotin. So both of them are synthesized by, in fact 3 of them are synthesized by the intestinal flora. Okay, one more vitamin is there which is synthesized by the body, that is vitamin D. We know that the precursor of vitamin D is skin, or you can say 7-dehydrocholesterol. Right? So these are the 2 categories of endogenously synthesized vitamins. So now first of all we are going to discuss, these are some of the differences or the important points. Now we are going to discuss first the fat soluble vitamins. So what all you have to know about a particular vitamin is, always keep one thing in mind, that these vitamins always, whether they are coenzymes or not, say if we are talking about fat soluble vitamins, they will carry out a biochemical reaction, they will be absorbed, they will undergo assimilation, and they will perform their functions. And if that particular function is not performed, it will give rise to the diseases. And there is always a recommended dietary allowance for all the vitamins. So we should know what is the active form of the vitamin in which it is acting in the body, how it is acting, how it is catalyzing the biochemical reactions, and if a particular biochemical reaction is not catalyzed, what diseases it leads to. So let's start up with the first vitamin, vitamin A. Now vitamin A is also known as, obviously just now I told you it's a fat soluble vitamin. And if we talk about the structure of vitamin A, it has a beta-alanine ring, plus isoprenoid unit. Right. And this beta iodine ring is actually present in beta-carotene. So we call these vitamin A, the other form is retinoids, which are having carotenoids as one of the basic structure. Okay. So if I'll just show you the structure of vitamin A. Let's take it to other page. Now, since I told you that vitamin A, the other name is retinoids, so there are basically three forms of vitamin A. Commonly we call them as retinoids. It's a class basically. So these retinoids will have retinol, retinal and retinoic acid. Retinol, ol mane alcohol, al mane aldehyde, oic means acid. So you can see that all the three forms are like they are related with each other. Whenever we oxidize an alcohol, it is converted into an aldehyde. Whenever we oxidize an aldehyde, it is converted into an acid. So these three forms are interconvertible with respect to one another, right? And this is the structure. If we talk about retinol, you can see that the OH group is there, right? Because retinol means alcohol is there. And if we talk about retinal, retinal means CHO is there. And we know that it can be, when it undergoes oxidation, it can be converted into retinal. And if it undergoes oxidation, it is converted into retinoic acid. Right? So these three are the group of the retinoids, will come under retinoids. And if we talk about the structure, they have a beta iodine ring which is attached to a isoprenoid unit. And if we talk about beta-carotene is having a iodine ring. And this beta-carotene is actually cleaved by a enzyme which is dioxygenase, and this reaction takes place in the liver. If you count the number of carbon atoms, it has 16 carbon atoms, and there will be an even splitting in the presence of the enzyme intestinal dioxygenase, and it is going to form retinal. So I can say 2 molecules of retinal. Right. And this retinal will be, since they are fat soluble vitamin, they will be taken over with the help of, the moment they will leave the intestine, they will be with the help of chylomicrons. Right? They will be taken over and stored in the liver, and that too in the perisinusoidal cells or the ITO cells of the liver. As you can say palmitoyl esters. Palmitoyl esters. Right? Now, because I told you this is the main difference between the fat soluble and the water soluble vitamins. Water soluble vitamins ka function is they will just act as a coenzyme and they will be excreted in urine. But fat soluble vitamins can be stored in the liver, and whenever their function is required, they will move to the target sites. So move to the target sites as retinal binding protein, transthyretin and retinoic acid binding protein, because they are fat soluble. The moment they enter into circulation, they will seek a carrier protein. As we have discussed, just like that we discussed in lipoproteins, you know, कि whenever any lipid molecule which is non-polar, whenever it has to move in circulation, it requires a carrier molecule, and that carrier is generally a protein. So here also you can say it is retinal binding protein, transthyretin and retinoic acid binding protein will be there, which will move to the target sites. Now the question is, what is the function of vitamin A, or what is the function of these three forms of vitamin A, because it is existing in three forms, so their function has to be a little bit different. So if we talk about retinol, because I told you there are three forms: retinol, retinal and retinoic acid. Right? So the function of retinol is it helps in reproduction. The function of retinol is it helps in reproduction. If we talk about retinal, it helps in vision. And if we talk about retinoic acid helps in the growth, morphogenesis and differentiation, cell differentiation. So you can see that all the three forms have different functions. So we can say that retinol helps in reproduction, retinal helps in vision, and retinoic acid helps in the growth, helps in the morphogenesis and the cell differentiation. So first of all, we are going to discuss about the function of retinal, which is very, very important, because we have studied also that vitamin A is good for eyes, isn't it? We say carrots are good for eyes. Why? Because carrots are having beta-carotene. And if we talk about the richest source of vitamin A, that is halibut oil. Halibut oil is the richest source of vitamin A. And we can say carrots. Why carrots? Because carrots are having beta-carotene, and beta-carotene can function as an antioxidant also. This is also a very interesting feature about vitamin A. So they are the richest sources. Right. Now we are going to discuss the function of vitamin A in vision in detail. Right. So let's see that role of vitamin A in vision. Now, we all know that the eyes are the ones to see. Now, in the eyes, there are two types of cells, rod cells and the cone cells. So, if we talk about the rod cells, the main function or the main difference between rod and cone cells is that rod cells help in night vision, right? And cone cells help in day vision. This is sometimes what happens is, I'll just give you one common example. Whenever we go and watch a movie, the moment we enter in a movie hall, cinema hall, for 1 second, sometimes for 2 seconds also, our eyes are unable to adjust. We cannot see even the seats where we are going to sit and watch the movie. But after some time, our eye acclimatizes and we can see the seat and we can watch the movie also. So what is happening in that particular interval, that the eyes are adjusting themselves so that we are able to see in the dark. Right. So there these rod cells will help. Suppose once the movie finishes, we have to come out. The moment we come out, sometimes you must have observed that, it's like a different type of light strikes our eyes. Sometimes we are unable to adjust ourselves. We cannot see also in the bright light whenever we are coming out from the dark light. So again, what is happening is the cone cells are releasing something which will help us to adapt our eyes to see in the bright light. So there is a transition phase, movement from the bright light to dim light, and again coming back from the dim light to the bright light. So these rod and cone cells will help in that. Now the question is, what is the role of vitamin A in that? So what happens here is that whenever, or you can say in the rod cells, there is a pigment which is known as rhodopsin. So this rhodopsin is made up of rhode and opsin. So basically, whenever we are entering from the bright light to dim light, this rhodopsin will immediately be generated. Now the question is, I told you retinal. So which form of retinal activates this rhodopsin and what is the role of a second messenger in this? You know, everything is interrelated. They are going to give a message to the second messengers that now this process has to be opened up. Okay. So what happens here is there is one 11-cis retinal. So this 11-cis retinal will activate rhodopsin. So I can say rhode plus opsin, they will combine and rhodopsin will be produced. Right? So I can say that from bright to dim light. Right? This rhodopsin is generated. Up now what happens is the function is over. This rhodopsin, the person comes out. So what we are doing is we are entering from the dim light to the bright light, dim light to bright light. Now the function of rhodopsin is over. That means it has to cleave. So this is cleaved into all trans retinal. Yeah, I can say that 11-cis retinal, right, it will help in the combination of rhode plus opsin forming rhodopsin, and this rhodopsin, when we enter from the dim light to the bright light, it is cleaved and it is cleaved to form all trans retinal. And here different combination of, you can say, pigments are there. If you have witnessed sometimes, you know, green light, yellow light, all lights will flash on the eye. So it will be like meta-rhodopsin. Rhodopsin is there, 𝗵𝗵rhodopsin is there, 𝗵𝗵rhodopsin is there, so many are there. Right. So all will convert this rhodopsin into all trans retinal. Now the function of 11-cis retinal is complete. Now this all trans retinal will then enter into the liver, and liver mein it is going to get converted into all trans retinol. Means retinal is converted into all. So obviously the enzyme here is dehydrogenase. NADH plus H positive NAD. And this all trans retinal will then be converted into 11-cis retinol. This is isomerase because cis and trans. And this 11-cis retinol will then be converted into 11-cis retinal by again dehydrogenation. So the enzyme is dehydrogenase. So I can say that this particular reaction is taking place in the eye, and this particular reaction is taking place in the liver. So I can say that dehydrogenases, which is converting all trans retinal to retinol, is present in the liver. Then by isomerase it will be converted into 11-cis retinol, which will then by dehydrogenation be converted into retinal. So I'll just highlight the groups here. Here it is retinal. Retinal. You can see here it is retinol, here it is retinol. Okay. So retinol. Retinol. So are you noticing the difference? Retinol, retinol. But isomerase, trans, cis. Retinal, retinal, cis, trans, dehydrogenase, and like this. So this is known as Wald's visual cycle. It was actually given by Wald. So we can call it as Wald's visual cycle. And what is the role of vitamin A? We can say that vitamin A has two forms, 11-cis-retinal and all-trans-retinal, which is converted into retinol, and this is how one is able to adjust himself when he enters from the bright light to the dim light and from the dim light to the bright light. Now the question is, I told you there is an involvement of a second messenger also. So which is the second messenger involved here? It is actually GPCR induced. So what happens here is that inactive GPCR, what is GPCR? It is simply G protein coupled receptor. It will be converted into active GPCR in the presence of beta-rhodopsin 2. So what happens is, when this inactive GPCR is converted into this G protein coupled receptor, right, it is activating transducin. Now what happens is, the inactive phosphodiesterase enzyme, inactive phosphodiesterase enzyme will be converted into active phosphodiesterase enzyme, and this active phosphodiesterase will convert cyclic GMP into 5' GMP. Matlab, you see how the rate of reactions is going on. First of all, you can say beta-rhodopsin is there, which is making GPCR active. When GPCR is active, it is accelerating the activity of transducin. This transducin is helping in making phosphodiesterase active. And we all know phosphodiesterase ka function. What is the function of this? This phosphodiesterase converts cyclic GMP into 5' GMP. And once this 5' GMP is released, it will close the sodium channels. And when it closes the sodium channels, it leads to hyperpolarization, right, thereby generating the nerve impulse. The nerve impulse, right. So this is how the entire chain goes. And this equation, I would just write the full equation. Actually, the equation which is working here is this. We know that GTP in the presence of guanylate cyclase forms cyclic GMP, which is this. And this cyclic GMP in the presence of phosphodiesterase enzyme forms 5' GMP. So once 5' GMP is there, it is going to close these sodium channels by leading to hyperpolarization and generation of the nerve impulse, right. So this is occurring stepwise. So this is one of the very important uses of vitamin A, and the active form of retinal, which is helping in generation of rhodopsin, is 11-cis-retinal. Once the function is over, 11-cis-retinal is converted into all-trans-retinal, and then the entire reaction takes place in the liver, because liver is the site for the storage of vitamin A. And then second is that this vitamin A is also required for the synthesis of the growth, retinoic acid, basically growth in cell differentiation and morphogenesis. or basically it is helping in the growth of the epithelial tissue, thereby epithelial tissue, right. And another function is obviously how in the growth of the epithelial tissue we can simply say that it interferes with gene expression also. Right. So these are the two very important functions, and I told you about retinol which helps in reproduction. So these are the important functions of vitamin A. Now there are certain other I would say like structural analogues which are resembling this carotene, but still they are not a component of this. So non-vitamin A carotenoids, they are, or you can say the structural analogues. So if we talk about there, जैसे lycopene is there. So lycopene is required for the treatment of prostate cancer. For the treatment of prostate cancer. Right. This is again, it is resembling, it is having a carotene type of structure, but it does not come under a vitamin A derivative. That is why I have written non-vitamin. Then lutein is there. Okay, then lutein is there, which is required for the treatment of macular degeneration. Clear? So you can say these are the non-vitamin A derivatives. After this, then we come over to these are the functions of vitamin A. Now what are the deficiency manifestations of vitamin A? Because vitamin A is not acting as a coenzyme. What are the deficiency manifestations of vitamin A? Now the earliest sign of vitamin A deficiency is the person will lose its sensitivity towards green light. So earliest sign is loss of sensitivity towards green light. Sensitivity towards green light. Right. And earliest symptom, if we talk about the earliest symptom, it is night blindness. Why? Because if the dark adaptation time is increased, the eyes are unable to generate rhodopsin. So earliest symptom is night blindness. And this night blindness is also known as nyctalopia. Right. Then we can say ki what happens is due to loss of sensitivity towards green light, night blindness due to improper growth and differentiation, what will it will lead to xeric conditions in the eye. So it will lead to the formation of Bitot spots. Foam cells will be there. And the reason behind this is, you can say xeric conditions will be there, which is also known as xerophthalmia. Overall, it is all under xerophthalmia only. Corneal vascularization is there, corneal dryness is there, corneal keratomalacia is there, so many things are there. So you can say overall this leads to xerophthalmia. Zero means xeric conditions in the eye. Xeric conditions of eye. Right, I'll just show you the formation of Bitot spots here. This is how the eye will look like. These are the Bitot spots, you can see that. Excessive dryness in the eye leading to corneal vascularization, corneal keratomalacia is also there. Right? So overall we refer it to as xerophthalmia. Then it is required for the growth of the epithelia, but again absence of this will lead to follicular hyperkeratosis. Right? And in fact it leads to what? It leads to pheroderma. You can see rough scaly skin is produced. The pheroderma, which is also known as a kind of toad skin. Okay, so we can say that deficiency manifestations of vitamin A are, what is the earliest sign? Earliest sign is loss of sensitivity towards green light. Earliest symptom is it leads to night blindness. Then you can say that xeric conditions are there in the eye leading to corneal vascularization, leading to corneal keratomalacia. Overall it will lead to the formation of the foam cells leading to Bitot spots, and we can call it as xerophthalmia. Second is vitamin A ki forms are required for the growth and cell differentiation also. So if it is not there, it will lead to you can see that it will lead to hyperkeratosis, follicular hyperkeratosis, and the individual will have a toad-like skin, which is also known as pheroderma. Pheroderma is a characteristic feature of deficiency of essential fatty acids also, because again fatty acid fats, fat-soluble vitamins, so they are requiring one agent for the transport because they are not soluble in water, right? So these are the deficiency manifestations of vitamin A, and I can say that due to dark adaptation time is increased. So if dark adaptation time is increased, rhodopsin is not generated. Is not generated. Right? So these are the deficiency manifestations of vitamin A. Then another very important feature about vitamin A is that if we have vitamin A in bulk, it will lead to hypervitaminosis. Because if we are eating vitamin A more than the RDA, it will lead to hypervitaminosis or vitamin A toxicity. So, vitamin A toxicity, or it is also known as hypervitaminosis. Right? Now what happens? There are actually only 3 vitamins, I would say, which are toxic. One is vitamin A in more doses, then vitamin B3 in more doses, and vitamin B6. They are the 3 toxic vitamins. So what happens here is, this hypervitaminosis is a characteristic feature of people who are Arctic explorers. Arctic explorers means because Arctic area, it is the coldest area, so they eat polar bear, and that too liver of the polar bear. Okay, and we know that vitamin A is stored in the liver. So obviously if they are continuously eating that liver of polar bear, they will have more intake of vitamin A, and the organelles which are damaged, they are the lysosomes. So you can say organelle damaged are the lysosomes. And what are the biochemical manifestations of vitamin A toxicity? What are the biochemical manifestations? right? It leads to pseudo-tumor cerebri, there is increase in the intracranial pressure. Increase intracranial pressure is there, right? Then it leads to basically hyperlipidemia or hypertriglyceridemia. Obviously liver में है, so the liver will be enlarged, so hepatomegaly will be there, right? And then the person, there is a loss of appetite also, loss of appetite, right? Loss of appetite will lead to again weight loss. Weight loss will be there, anorexia will be there, right? So I can say that vitamin A toxicity will lead to pseudo-tumor cerebri, hyperlipidemia, hepatomegaly, loss of appetite, anorexia and alopecia also sometimes. So in some patients alopecia has also been observed. There is loss of hair. Okay. So this is vitamin A. And then we come over to how the assay of vitamin A is carried out. So whenever we have to carry out the assay of vitamin A, first is serum determination, serum estimation of vitamin A. By Carr and Price reaction. And second is by evaluating or measuring the dark adaptation time. Right. So this is vitamin A. Very, very important fat soluble vitamin whose active forms are retinol, retinal, retinoic acid. The function of retinol is reproduction, retinal it plays a very, very important role in the vision, and retinoic acid which plays a very important role in growth and the morphogenesis. And if we talk about the role of vitamin A in vision, I told you the most important point which you have to remember is its active form, which is 11-cis retinal. The moment we enter from a bright light to a dim light, or example is theta, so rhodopsin is generated. So it helps our eye to focus. Then the moment we come out, this rhodopsin is cleaved to form all transretinal. The function is over. This all transretinal will be converted into retinol. This retinol will be converted into its own isomer, which is 11-cis retinol, and then again it will generate 11-cis retinal. And the most important point which we have to remember here is that vitamin A is stored in the liver. That is what I have written here also. Storage is always in the liver. And where in the liver? In the ITO cells of liver. And whenever vitamin A is required, automatically it will move to the target sites. The thing is that whenever vitamin A has to move, it has to search for a protein. So it will be RBP, retinal binding protein, transthyretin and retinoic acid binding protein, and it will be carried to the target organs. And there it is going to exert the function. Now if vitamin A is deficient, what are the deficiency manifestations and how it acts in the opening and closing, that is with the help of G protein coupled receptor. G protein coupled receptor is made active by meta-rhodopsin. Meta-rhodopsin is the form which is produced when rhodopsin is being cleaved into all transretinal. Okay, so this will activate transducin. Right, this is active basically. Then inactive phosphodiesterase will be converted into active phosphodiesterase, and then 5' GMP will be produced because phosphodiesterase is the enzyme which always accelerates the conversion of cyclic GMP into 5' GMP. So it will close sodium channels leading to hyperpolarization, and it helps in the generation of the nerve impulse, right? And then second is, this is the function you can say, and then deficiency mein the earliest sign of vitamin A deficiency is always loss of sensitivity towards green light. This is very important, okay? And then the symptom is, when dark adaptation time is increased, it is night blindness. Then it will lead to the formation of Bitot spots, and overall it will be known as 𝗲𝗵𝗵𝗺. The eye will undergo xeric conditions, right? Corneal keratinization will be there like that, and the skin will become scaly, very rough skin will be there, which resembles the skin of a toad, and that is why we say it is known as pheroderma or the toad skin. Then the question is, how do we assay? So how do we take the assay? Before assay, yes, this is very important. Vitamin A. There is a recommended dietary allowance for every vitamin. And I told you there are 3 vitamins basically which are toxic if we increase the RDA. One is vitamin A, second is vitamin B3, and the third is vitamin B6. Now, why vitamin A becomes toxic? That people who are residing in the Arctic areas are very cold areas. So, their mode of survival is just the meat, and that too they eat the liver of the polar bear, because polar bear is found there. So, it will lead to what? It will lead to increase in the intracranial pressure, which is 𝗲𝗵𝗵𝗺 cerebri there, right? And obviously, if liver we are having, so it will lead to hypertriglyceridemia. There will be enlargement, it will lead to hepatomegaly, it will lead to loss of appetite, weight loss, alopecia. So this is what? This is vitamin A toxicity, which we refer to as also hypervitaminosis, right? And the organelles which are damaged are the lysosomes. Very important again, right? And then how we carry out the assay of vitamin A? Simple serum estimation by Carr and Price reaction, and we can measure the dark adaptation time. So this is all about vitamin A, which is a very important fat soluble vitamin, or whose main role, whenever we talk about vitamin A, the main thing which should come to our mind is the visual cycle. It helps in vision. And if we talk about the richest sources of vitamin A, they are the halibut oil and the carrots. Why carrots? Because carrots are actually rich in beta-carotene. And I have told you vitamin A ka structure, if we see, it has a beta-iodine ring which is attached to a isoprenoid unit. So, the beta-iodine ring hai, it is the same which is present in carotene. So, carotene is performing the functions. So, I hope this vitamin A video is clear to all of you. The deficiency manifestations, the functions, everything. Right? So, this is all in this video. Thank you so much.